You know, it was a thermal cycler that does this thing. The temperature will be reduced to 55 degrees centigrade, where primer will be added to it. That was called aniline. And then each new different, DNA strand will be formed. Okay. And the third step is extension. The extension means that the new strand will be synthesized, and the temperature is slightly increased from 55 degrees centigrade to 72 degrees centigrade. All what I'm explaining, you can see in this diagram. You can see the DNA sample, you can see the primer, you can see the nucleotides, you can see the tag polymerase, mixed buffer and the PCR tube. So those are exactly that are the component of the PCR. You can be able to draw the diagram of this PCR component is to give you clear understanding of what we discuss here. And there's a video clip I show during the time of lecture, about 2 video clip I show for us to see how does this thing work. How does it work? If you go one time, 2 times, or how many DNA will you intend to produce? This is the note. So another thing is that you need to know what does the primer, what does it do? And remember that I say primer is just like a kick. You want to start a car, you put a key inside the car, and it's a kick that initiate the ignition of the car, that start the car because before the car can move. Primer is a DNA synthesis. The primer is a short single-stranded segment of nucleotides of RNA or either DNA that provide a starting point for DNA replication. For the DNA to replicate, to start being synthesized, it's a primer has to come in contact. And what is the function of this primer? Primer is essential for DNA polymerase enzyme, which is synthesize new DNA strands. DNA polymerase can only add nucleotide to an existing strand. So primer provides the initial 3-hydroxy group to which new nucleotide can be added. Primer is usually about 18 to 24 nucleotide, long and designed to be complemented to a specific sequence. A question can come, write short note on primer. Write short note on primer. Now you define what is a primer. You tell me what the function of primer, and then the composition of primer, and you get your full mark. Talked about how primer can be designed. We talked about initial denaturation. Those 3 items I talked about are mainly, the initial denaturation, that is from 94 to 98 degrees centigrade. Why the hydrogen bond between the DNA, they begin to break down to pieces, and then it happened, and then the temperature is lower to 50 to 65 degrees centigrade to allow the primer to bind RNA to their complementary sequences. I explain and I show you the video. If you could not remember, go to the video, click on the video and see. And then extension. Before the circle, the circle that is replicated. Pay attention to this circle repetition. Pay attention to this circle repetition. I said these steps, denaturization, annealing, and extension, are repeated 20 to 40 times. Cool. With each cycle, the amount of target DNA double, leading to exponential application of desired DNA. You must know if that's the cycle, the first cycle, how many DNA is going to be produced. If you go for 2-3 cycles, how many DNA is going to be produced? But it should be, there's a cycle repetition. Pay attention to the cycle repetition of each of the DNA, primer. I started some example as it is shown on the screen. Initial denaturation, linear, and extension. Now, another thing, application in the lab. What are the applications? You want to look at the application of this PCR in the laboratory diagnosis. Number 1, infectious disease detection. we all remember during COVID-19, most of the laboratories, there's no availability of this polycycler, So they need to get to purchase a lot of equipment for them to get this sample detected to be diagnosed. Then federal government spend hell of money. So what does it do? One of the applications is to infectious disease detection. It's one of the best way to detect infectious diseases. Number 2, genetic disorder. We all heard of sickle cell anemia, fibrosis, cystic fibrosis, and sickle cell anemia. The genetic mutation of various abnormalities in the body systems. can be, can be screened, and can be diagnosed to know where the mutation really occur. This PCR can do that. Also, cancer diagnosis, PCR can detect specific genetic changes associated with different type of cancer, aiding in early diagnosis and personalized treatment plans. Also, in forensic science, in forensic laboratory, PCR is used to amplify DNA from crime scenes. Sample for identification purposes, it helps in paternity testing, criminal investigation, and disaster victim identification. In forensic medicine, for example, somebody died now. The best part of the blood, the body can get the accurate sample is the aqueous humor in the eyes. It doesn't die. After maybe between 24 hours of death, you can pick those samples and use it to analyze. Use to analyze and detect what really the cause of the death of such an individual. Number 5. Who is sending a message? Number 5, it can be used. Well, sorry. Somebody sent me a message. I think it's dropped my screen. I can't see the screen again. What happened?
I'm seeing the screen, sir.
We are seeing it.
Can't see from my end. Okay, wait.
Yeah, please.
Okay. Then number 5, PCR is fundamental tool in molecular biological research. Like for example, when I was doing my PhD, the PCR I use PCR to do my analysis in molecular biology. Most of the molecular biologists laboratory, they have a PCR machine for research. I enable scientists to study gene expression, clone gene, and conduct various experiments. DNA structure, the component of DNA structure, DNA replications, then summary. Don't bother yourself with a stage of gene expression. Okay. RNA processing, translation, initiation, elongation. Don't stress yourself concerning that one. It's not necessary in these coming exams. Thank you. Then let's go to the next one. Nucleic acid. All right. Can you see the screen I shared? 413. Hello?
Yes, sir.
All right.
Yes, sir.
So 413, we pay attention to this this topic. Nomenclature for basis nucleoside and nucleotides. The base or the basis of DNA or RNA. They are what we call a building block of nucleic acid. They're equally called the bases, which are adenine, guanine, cytosine, thymine. That thymine can only be found in DNA, while uracil replaces thymine in only RNA. I repeat again. In DNA, we have adenine, guanine, cytosine, and thymine. While in RNA, we have adenine, guanine, cytosine, and uracil. That means uracil replaces thymine in DNA. That is the building block, the basis. Pay attention to that. All these things will come out in the objective MCQ, multiple choice questions. And we talked about a nucleoside. And I said a nucleoside consists of a base linked to a sugar. These those bases we mentioned, adenine, guanine, cytidine, they link to a sugar in bracket, ribose in RNA and deoxyribose in DNA. That's nucleoside. The basis, a nucleoside consists of a base linked to a sugar. That means the base, they link up with a sugar compound. Ribose, if it is in ribonucleic acid, that is ribose. While the deoxyribonucleic acid is deoxyribose. The names of nucleosides are derived from the bases. For example, adenosine. That means adenine plus ribose together give us adenosine. While guanosine is combination of guanine plus ribose. While cysteine is cytosine plus ribose. We have thymidine, that is thymine with deoxyribose, that is DNA. And we have uridine, yeah, uracil and ribose. Be able to know difference between the bases combination of one or two bases form nucleoside. A combination of one or two nucleosides with sugar give us what we call nucleotides. The nucleotide is a nucleoside with one or more phosphate group. The only difference is that the nucleotide is that after the addition of the sugar link of sugar compound to the bases, here in nucleotide, there is still additional of phosphate group attached to the nucleosides. And the name based on the nucleoside and the number of phosphates. We have adenosine monophosphate, we have adenosine diphosphate, adenosine transphosphate. For example, I can say write short note on nucleoside and nucleotide. Be able to define what is a nucleotide. Be able to cite an example, 3 to 4 examples of different type of nucleoside or nucleotide. Okay. All right. Then we have nucleic acid and the hydrolysis. Nucleic acid are biopolymers, essential to all known form of life. We have DNA, we have RNA. I want to look at structure of nucleic acid. I talk about sugar phosphate backbone. A chain sugar ribose in the RNA and Y deoxyribose in the DNA and phosphate group. We have nitrogenous bases. The base, as you mentioned, guanine, cytosine, thymine, and uracil replaces thymine, DNA. Let's say hydrolysis. Hydrolysis breakdown. We have enzymatic hydrolysis, we have acidic hydrolysis, we have alkaline hydrolysis, all those things. Please pay attention and go through them. Now, we want to look at DNA sequencing definition. What is DNA sequencing? DNA sequencing is the process of determining the sequence of nucleotide within a DNA molecules. Every organism DNA consists of unique sequence of nucleotides. Determining the sequence can help scientists compare DNA between organisms, which can help show how the organisms are related. Look at overview from what you mentioned. Talked about the bases, guanine, thymine. Talked about modern sequencing consists of high thorough method that allows entire DNA sequence to be discovered in a matter of hours. Application in disease, in disease diagnosis. Pay attention. Genetic, like I told you earlier now, genetic disorder, cancer diagnosis, infectious disease, pharmacogenomic, prenatal testing, and for research. So, and like I said, the MCQ, it contains everything in this particular topics. Everything you note, it contains. So pay rapt attention, read over and over again, and I can assure you that you come out in flying colors. Okay? And this come to the end of the class. Do you have any question? Any question?
Thank you very much, sir.
Any comments, observation, or anything that bothers you that you are afraid of? So feel free and ask questions. I think all of you are going to my place to write the exam, right?
Yes, sir.
Yes, sir.
I think I should be around. I should be around that time. I'll be on leave, and so I hope to be maybe 1 or 2 days. Especially the day they want to write my course, I will strive to be around. I need to be able to identify. I don't know most of the students here. If I met 1 or 2% in Lagos and I couldn't identify anybody, I'll be away for a long time. So that would be an opportunity to know the student physically, and then we take it up from there. And shortly after your exams, I don't think there's any break whatsoever. It depends on the HOD. You prepare to do, you are going to specialize in each specialties. Those who want to go to chem part, medical microbiology, histopathology, cytopathology, whatever. And as a room for change, if you do chem part before you want to change to anything, you are very free. All what you need to do is just to write your project and then pay attention to those. And then you'll be presenting seminar first semester. You present a seminar. You get a seminar topic now. You're going to do a presentation. If I were you, I would prefer to take choose a topic I want to use for your project. You are going to do project, and project is 6 credit units. Project is 6 credit units. Now, what that means is that if you can have an exciting and good, if you have A in your project, you can easily have first class. So, and if you now after your exams, don't look for a topic that's convenient. Choose a topic. You are free, liable to. I think they will share you choose your supervisor, choose your topic. Like those people that want to work with me, I don't give a topic to students because I want my student to choose their topic of choice. When you choose your topic, you'll be able to flow. It's your interest that will pushing you in case if there's any challenge along the line when you are doing the project. So prepare for the seminar, presentation. Second semester also you are going to do seminar too. Then before you defend your project. By this time next year, we should be open. We'll have rounded up and be preparing for graduation by God's grace. If things work, all things be equal. Thank you.
Thank you, sir.
Thank you very much, sir.
Any questions?
Please, you've explained much on PCR, but sir, especially about the diagram, if you can please explain more in a way that we still understand it better. I will appreciate that, sir.
I just when I talked about the PCR, I can project again. When I talked about the PCR, the principle, there are major key steps that is very important in PCR. You talked about denaturization. What I mean by the double-stranded DNA is being heated up. This thing is done in a machine, the equipment, this thermocycler, that's what they do. I don't know how many of you have seen it before. So automatically it increases the temperature, and the DNA strand will expand and break into smaller units. That's what they call denaturization, when it is denatured, And the second step is annealing. And what annealing means is that primer will now come into place. Primer is just like a key. You want to move a car, you need to put your ignition into the car, you start. So that's what primer does. What primer does, and there's a video, if you have that, check the platform, the video, just watch that video, pay attention and watch that video. I can see when the primer is being added, the strand begin to elongate, it increases in size, and it break up into another, DNA.
Okay, sir.
So when the primer is, attached to, when primer gets there, it expands. That expansion is called, extension. The temperature will decrease again. The temperature from the 95 to 75 or 76, from 95 to 56, the temperature was reduced where the primer is being added. When primer is being added, the temperature will be increased again for extension to take place. I don't know whether you're getting my point.
Yes, I'm getting it.
So if you can be able to explain this point properly, it's as simple as just 3 steps: denaturization, annealing, and extension. And the key thing in the denaturization must mention the accurate temperature that the denaturization happened. And then annealing, at what temperature does annealing happened? And the third one, extension, at what temperatures does it happen? Before you now talk about the circle, repeated circle. It's as simple as that. For you to remember the diagram I showed the other time, thermocycler, DNA, primer, whatever, you can sketch it and draw it. So when you put on your note, you can be able to remember as much as possible.
All right, sir.
If you draw it also, it's additional mark to you. So it can to help you to navigate through the PCR method. I must know, as I said, what are the uses, what is the importance of DNA? Of the PCR. What does it do? Which area can you use it?
Yes, sir.
So I hope I've explained myself.
Yes, sir. Thank you very much, sir.
Or before your exams, you can go to any laboratory. There's some laboratory that have a PCR machine. Go there, just look at it and then see. Maybe run the sample. You can see it by yourself, and then you can take it out from there.
Yes, sir. Thank you, sir. God bless.
Any comment? Some people, they keep quiet. They don't talk. They won't talk in the class. Feel free. Don't come and chat me up or call me behind the scene or call me, talk to now. Let everybody hear you out. Don't chat me up. Don't call me. If you cannot talk in the class now, don't ever. If you come, I won't pick your call. I save everybody with my person. I will not pick your call. So if you have any comment now, please. Yes, go ahead.
Hello. This is Rukayat. This is Rukayat.
Yes, I can see you and I hear you loud and clear.
Yes, sir. Please temper justice with mercy. You know, we have read for a long time. We have not been for almost 30 years now. So it's not easy reading all these courses together. Temper justice with mercy when you are marking us.
No, I've told you, nobody, I can assure you, I'm a man of my, those people that knows me, I'm a man of my word. The question is so said in such a way that nobody design and said in such a way, nobody, even if you don't read, have you listened to me this afternoon, you will not fail.
Thank you, sir. You are appreciated.
So what you need to do is just to believe in yourself, believe that you're going to pass. Nothing, no big deals. I made it such a way that it's as simple as what you can think of. I'm telling you. Class rep, anything to say?
Oh, I'm okay, sir.
Are you sure you're okay?
Thank you.
I told you people I don't give area of consultation. So I have to give area of consultation. I don't.
No, this one is all right.
My co-lecturer, they fought me, say, Doctor, give me half the area of consultation. I said, no, I don't give it up. But everybody, there's no problem. Everybody, I can assure you, you can hold me by my word. Everybody will do well. We do well.
Don't believe in the diffusion or osmosis.
You will do well. The MCQ question is so simple that it's all direct. 20 marks.
Sir, can you go over 406 again, sir? Hello, sir.
You say?
Thank you, sir.
No, sir.
Go ahead. I can hear you loud and clear. Come again. I can't hear you now.
Sir, can you just go over 406 again, sir?
You came late to the class?
Yes, I joined. I actually joined on spectrophotometry. I joined when you were on photometry. So what did you do before photometry?
Once you come late to my class, in fact, if it is a regular class, you will not even, I won't allow you to come in. So it's a sign of, for you not come early, I won't repeat. I don't repeat myself.
So
my
sorry,
class,
sir.
natural class, if I'm fixing a lecture by 11 o'clock, it's expected to be around 5 to 10 minutes before the exam, before the lecture. So for coming late, I will not repeat myself. For coming late.
Okay, thank
you.
You,
Read
sir.
your note. It's there.
Thank you.
So in absence of no other question, no comments, can we
excellent lecturer,
call it a day?
Excellent lecturer.
We are very grateful, sir.
Thank you, sir.
Thank you very much.
See you on
yes, sir.
See you on 13 of August, 15 of August, and it's 12 of August, 13 and 15 of August.
By God's grace, we shall meet.
And those people that submitted their assignment is well acknowledged. Some people say I didn't respond. I acknowledge they have marked it and I've scored it for those who that submitted.
Okay, sir.
Thank you, sir.
Thank you very much.
God bless you. Do have a wonderful day.
Sir. Thank you,
You
sir.
too.
God bless
Goodbye.
you too, sir. Thank you, sir.
Thank you so much, sir. Please, let's go on. If we have any complaint about the portal, please fill that form I shared on the. Yes. So concerning the second form, once I am through talking, once I'm through talking to the HOD, I'll send that to that form.
Okay. All right. Thank you.
I've filled the form already.
Yeah, I will fill it now.
Mr. Fasasi. My friend, Mr.
Yes.
Fasasi. I chatted you up to send something to me. You haven't.
Because I am busy. I'm and
okay.
I'm not
okay. You
there
say
yet,
you say so. You say you're on call.
My is that I'm even attending.
Network.
That I'm attending to anybody because I'm just trying to. I'm not still okay. That's just the point. Sir. I would like to keep things together. So please, let's stress on me. Thank you.
Okay, no wahala.
Send me your email. I'm not going to send anything to anybody over WhatsApp.
Okay, sir. Okay, sir. Are we still having an evening revision class? 6 p.m.
Yes, now.
Okay,
the lecturer has not said. So let's do our own. We'll communicate us. Yes.
Mr. Lalekan, we can see your laboratory.